Hello, everyone. My name is Jolla Abshier. I am a second-year PhD student at Portland State University, working in the Center for Life in Extreme Environments, more specifically out of the Extreme Virus Lab. And today I'll be giving you a little bit of insight into life in hell, understanding the role of toxin-antitoxin systems in prokaryotic genomes and their potential for virus-host coevolution, TLDR, extreme organisms, the viruses that infect them, and their interactions in these extreme environments. And our journey kind of begins right here. So this is a personal image of Lassen Volcanic National Park. It is one of the many areas on our beautiful planet that is home to these volcanic hot springs. This is one of the main sampling sites of our lab. And while most people wouldn't believe that there are things living in this environment, there certainly are. Specifically these microbes from hell. And I'll get back to these guys in just a second. But a little bit of background about why we study these particular environments. So these volcanic hot springs are often considered and understood to be analogs for planets both outside and inside of our solar system, referring back to some of the ancient hot springs found recently on Mars, which may or may not have looked something like this boiling mud pot that we find in Devil's Kitchen at Lassen Volcanic National Park. Getting back to the microbes, one such microbe is this microbe right here, a spherical cell known as Saccharolobus olfactoricus. This is a scanning electron microscope showing you the topology of the cells. Coined S441 by our lab, really thrives and lives in these hot acidic conditions with temperatures from around 75 to 80 degrees C and pHs of around 3 to as low as 1. So these really extreme environments. And when we think about these environments and the rich ecosystems that habitat them, we can also think about one of the most abundant molecules on the planet, so that of viruses. And yes, there are these extreme viruses infecting these extremophiles. The one which I work with in lab is Sulfolobus spindle-shaped virus 1. Saccharolobus is a natural host of this particular virus, this SSV1. This is Stanley, uses they, them pronouns, really characterized by his lemon shape. So these shapes are really unique to both archaeal organisms, extremophiles, and some of these extreme viruses. And when we think about looking at these organisms and the interactions between them in these extreme environments, we can look at some systems currently prevalent today in bacterial cells. So these toxin-antitoxin systems, usually known as addiction modules, found on plasmids, again, really prevalent in bacterial genomes, only recently being discovered to have some viral encoding. And what this particular system confers is when you have a plasmid that has the positive, or is positive for that particular addiction module, that cell needs that plasmid in order to continue its life in that particular environment. So here we have the presence of the TA. Should that cell or progeny continue with the presence of that plasmid, you would expect normal growth in the environment. Whereas if it were to lose that plasmid, it would have been addicted to that particular genome, it would die off. Whereas in a negative system, you would get growth either way, whether or not there's plasmid loss. And we can further visualize this particular mechanism by looking at what might happen to some uncolonized cells. Some sort of event happens in which the addiction module is introduced to that population. And you have your addicted survivor going on to make new progeny, and then conferring that group protection and persistence. So these systems are really understood in bacteria to confer a microbial persistence phenotype. Some have been referred to as phage defense mechanisms and largely considered as plasmid stabilizers on the plasmid. And we can also assume that another group of uncolonized cells were to come in here, perhaps that group encounter, you would still get that toxic culling from that toxin-antitoxin system. One important note, they are characterized as two genes that are typically right next to each other. The toxin being just downstream of the antitoxin, pretty stable toxin, pretty unstable antitoxin. And looking at this system with SSV1, a previous student did quite a bit of work in mutagenesis of the SSV1 genome. And we use this, at least these mutants, to test whether or not some of these genes or open reading frames are essential to the virus. And two genes in particular, this T3 and TX transcript, these particular two genes, we can make changes to what I think is the antitoxin, and we don't see too much difference in terms of viral infection, viral function. Whereas if we were to delete this or insert a sequence into this particular open reading frame, what I think is the toxin, we do see differences in infection mechanisms. And looking at infection mechanisms of these extreme viruses and just virus reproduction overall, viruses can typically go through two pathways or a combination of both. So a lytic cycle in which the viral genome, the viral DNA will insert itself into the cell. You have some replication going on, eventually assembly of those virions, and the cell will then burst, die, and those viruses will go on to infect other cells. Whereas in a lysogenic cycle, the viral genome is incorporated into the host genome, enters usually a latent phase in which it kind of just stays there, maybe some induction event happens. In SSV's case, it actually just buds from the cell without killing the cell. And one of the two ways that we use to test for this viral reproduction is, of course, PCR, so amplifying a sequence that's specific to viral DNA, looking for its presence in cell-free supernatants. Another way is through halo assay, so very similar to a plaque assay in which you look where cells are bursting. We cultivate a lawn of cells, uninfected Saccharolobus cells like you saw on the slide a while back. And then we spot cell-free supernatants, which contain these mutant viruses, onto the plate and look for clearings. And this halo assay is made possible because of the fact that SSV1 buds from its cell without killing the cell. And the infected cells grow quite slower, so it really confers this growth stunting phenotype in the Saccharolobus cell. In looking at the toxin protein, and a large majority of my work has been looking at mutants that we've made in this toxin protein, and several machine learning softwares have indicated that there's quite a high probability of a cleavage site over here behind this very hydrophobic helice right here. And so I sought to characterize at least this particular mechanism in SSV1 through using some of these mutants that we already have. And while largely these over here are wild type, so really shouldn't expect any kind of change in viral function, you can see some really nice halos, and we can confer that the cells are not dying, but they're sick. And in our mutants where we have insertions in that specific toxin open reading frame, we don't see any halo formation. Likewise, I sought to substitute those two residues at the cleavage site, since it's largely important for protein maturation, protein function, and we still don't see a halo just switching those two residues. But the main takeaway from this is that we still get virus replication. We still get quite a bit of at least high levels of viral replication from some of these mutants. In one of these mutants in particular, we didn't see anything, so maybe that one's detrimental to that particular open reading frame. But we do see that there is virus replication happening in the cell supernatant when we screen for DNA. And it's important to kind of note that this protein isn't just unique to SSV1. In looking at these systems in these extreme organisms, this one in particular up here is another SSV, so another spindle-shaped virus, in which we see really low sequence similarity across these particular genes, but quite a high bit of structural similarity between these genes. And a lot of these are genomes of extremophiles, while some are virally encoded. And one of the main takeaways, really just wrapping it all back around, looking for these particular mechanisms and how they confer persistence with both their extremophilic organism and their viruses would be a really nice first start in looking for these biosignatures, perhaps, and ways that we can look into how the microbes might be interacting in these particular extreme environments. This is another sampling video just of Lassen Volcanic Park, pretty close to where S441 was isolated from. Looks pretty similar to maybe something we'd see on another planet that we could sample at. And then shameless plug, but looking for viruses in space, I think, is the obvious next step. We know that they're the most abundant biomolecule on the planet, and just understanding that these mechanisms and that these systems are out there allowing these cells to thrive in this particular environment would be a great starter conversation for looking for these small biomolecules in space. And with that, I just want to thank my lab, my group over here at Boiling Springs Lake when we collected samples, and my PI, Dr. Ken Stedman. Thank you.
Thank you, Jono. We have time for about 3 questions.
Hey, my name's Pia, and I'm really interested in like CRISPR-Cas immunity of microbes. Have you looked at all into the interactions of other phage defense systems?
Yeah, so at least in terms of this toxin-antitoxin system, they're pretty understood in bacterial cells to like incorporate themselves into the genome as CRISPR-Cas9. There are some proteins that that look pretty similar to this A291 and some of these other virally encoded genes that do have some kind of CRISPR-Cas editing mechanism. So yeah, I think it might be leaning towards that way, but they're pretty well characterized in terms of like how they work with proteins and mRNA. So yeah, not too far there yet, but definitely somewhere in there.
Thank you for the talk. Marshall Seaton, JPL. And I was curious, so you're talking about looking for viruses in extraterrestrial environments. Do you think there would be, or I'm curious, I'm completely ignorant.
Yeah,
on the subject.
no.
I dropped out of bio too. So for this, for viruses specifically, like would small molecule biosignature classes vary from viruses to like what you'd be looking for with cells?
Yeah,
Or-
yeah, I for sure. And that kind of brings me at least back to a point like of looking for fossils and stuff like that. So viruses in sediment. And at least our lab, there's been some work in like silica encoding viruses and how, you know, the stability of them. So maybe there's a pretty silica-rich environment in which we might be able to look for any kind of these signatures. So like proteins or yeah, just specific things that might have been preserved in those particular environments. Considering that these viruses live and thrive in pretty extremes, you know, they got to be out there, at least in my mind.
And so you mentioned looking at like morphology and fossils and things like that. I know that at least for looking at cells and things, they've been shown to mimic abiotic systems pretty well because humans are very good at pattern recognition. And so has anything been seen like that for viruses? If you're mentioning fossils?
Yeah, not that I'm familiar with. I know it's at least a brand new conversation of starting to look for like viruses in space, astrobiology, things like that. So yeah, we're just kind of getting up and running.
Oh, yeah, no, sorry. I don't mean to like play 20 questions.
Oh, no.
I just thought it was cool.
You're totally fine.
Thank you.
Yeah. There's a little cure lab that like goes on right now that I teach, so I'm obviously not there, but there in spirit.
Any other questions?